So hello all of you. Today I would like to talk about different physical and chemical properties of amino acids. So let's first start with physical properties of amino acids. So in my previous class I have already discussed about these different physical properties of amino acids. Now in this class I would like to talk about two important physical properties of amino acid, that is amino acids are amphoteric in nature and amino acids act as buffer and has buffering capacity. So let's first discuss about the amphoteric nature of amino acid, which means that it can act both as an acid and base. So amino acid has two ionizable groups. First is carboxyl group and second is amino group, right? And here COH is more easily dissociable than NH3 plus group, and at neutral pH both groups are ionizable. So here at neutral pH COH is present at dissociable form and NH3 remain as associatable form. So you can see, you can easily notice here that NH3 has not been changed, NH3 plus has not been changed in at neutral pH. So this doubly charged molecule of amino acid is known as Zwitter ion, or it can also be known as dipolar ion or dihybrid ion, and it is electrically neutral, so it does not move in response to the application of electric field. The other two forms are anionic form and cationic form of amino acid, and the charge of amino acid always depends on the pH of the surrounding, and this property is generally exploited in the separation of amino acids. So now I would like to talk about how amino acids act as buffer. So buffer generally resist any change in pH value after adding a small amount of strong acid and strong base in an aqueous solution. So for understanding the buffering capacity of amino acid, you will have to understand the titration of titration of amino acid, and for that you will have to revise my previous class in which I have taught you about the titration curve of amino acid. So the acetic acid is the weak acid and its conjugate base generally act as buffer. So this is the weak acid, as I told you earlier, and it is going to be titrated with a strong base, that is NaOH. And at the beginning of the titration only the acid form present, and as you raise the pH by adding a strong base, that is NaOH, you will get a point where pH becomes equal to pK, right? And as you remember from Henderson-Hasselbalch equation, that pH only becomes equal to pK when concentration of weak acid and its conjugate base becomes equal, right? And here the pH, here at pH 4.76 the concentration of weak acid and its conjugate base becomes equal. So that's why this pH is known as pK. So now at around this point, when you will add a small amount of acid or base, this will not allow to change the pH of its aqueous solution. So in other words, in this region, any addition of small amount of weak acid and a small base generally resist any change in its pH value. So this whole region resembles its buffering region. Now if you will again add more NaOH, then a point will reach where all acetic acid converts into its conjugate base, and this point is known as point of equivalence or equivalence point. This titration curve resembles the titration of one functional group, that is carboxylic group, and in amino acid you will find two titrable group. That is, first is carboxyl group and second is amino group, and that's why during the titration of uncharged amino acid you will find two pKa value, one for carboxyl group and second for the ionization of amino group. So when an amino acid like glycine, which has one carboxylic group and one amine group, is in an aqueous solution, then the ionization of this amino acid will change with the change in the pH of solution. So how it will change? Let's understand it like this. Suppose in first case the pH of aqueous solution where uncharged glycine is present is very low. So low pH means high concentration of H plus ion. In this condition the amino acid prevails as diprotic form. Here diprotic ion means that this amino acid has the capacity to donate 2 H plus ion in aqueous solution. So in this case net charge of the amino acid is positive. Why it happened? Since H plus concentration was more, right, and more H plus will make bond with this amine group and will form NH3 plus group. Now if we further increase the pH of solution, then H plus concentration will decrease like this, and in that case COH group will lose one proton and changes into COO minus group in this amino acid. So this amino acid is now monoprotic ion because it has the ability to donate only one proton, and that is from NH3 plus group. And since this form of amino acid has only one negative charge and one positive charge, that's why the net charge of this form of amino acid is 0. And as I told you earlier that those amino acid which generally has net charge 0 is known as Zwitter ion. Now if we further increase the concentration of NaOH, then again the pH will increase and H plus concentration will decrease, and in that case this amino proton, this NH3 plus group will release one proton and converts into NH2 group, and in that case the Zwitter ion will convert into negatively charged amino acid. So by this way you have seen that how changing in pH from low to high, the positively charged amino acid converts into negatively charged amino acid. So by this way titration of amino acid generally happens. So to study this we draw a titration curve, and this is titration curve, and here question comes, what is titration curve? So as I told you in my previous class that a titration curve is a plot of the pH of the analyte solution versus the volume of titrant added as the titration progresses. So what is the analyte solution here? So here analyte solution is glycine, and what is the titrant here? Here titrant is NaOH. So in y-axis we have drawn pH, and in x-axis we have drawn OH concentration, which is increasing as we are adding more NaOH in the analyte solution. So as OH concentration is increasing, which means that H plus ion is decreasing. So we have already studied in our previous slide, as the pH of aqueous solution increases, the positively charged amino acid converts into negatively charged amino acid. So here as the OH concentration is increasing by adding NaOH, the concentration of H plus ion is decreasing in aqueous solution with amino acid. This whole process is shown by this curve. In this curve there are two phases. First phase showing a point that is pK1 value, and second phase is showing a point that is pK2 value. It means when amino acid move from positively charged to Zwitter ion, the dissociation constant is pK1, and when it moves from Zwitter ion to negatively charged amino acid, then the dissociation constant is pK2. So the pK1 is the pH at which the concentration of positively charged amino acid is equal to its Zwitter ion. So at this zone, when you will add a small amount of acid like HCl, first HCl will split into H plus and Cl minus, and by this H plus ion concentration has been increased. So to maintain the H plus concentration, this reaction will proceed in reverse direction, and by this way, at this region, the equal concentration of weak acid and its conjugate base will resist any change in pH value when in response to the addition of a small amount of strong acid. So this region generally shows the buffering region, right? So similarly, when you will add a small amount of strong acid base, then NaOH will split into Na plus and OH minus, and OH minus will react with H plus and will form H2O, and H2O will release in the aqueous solution. By this way, in this region, X plus concentration will decrease, and again to maintain this H plus concentration in the reaction will proceed in forward direction, and by this way this equivalent concentration of weak acid and its conjugate base will resist any change in the pH value at this zone in response to the addition of strong base, right? So around this phase, that is pK1 is equal to 2.34, glycine acts as buffer. So now when you further increase the concentration of NaOH, all the amino acid molecule will convert into Zwitter ion, and at this point only Zwitter ion present in aqueous solution. And as you know, Zwitter ion are those ionized species which have net charge 0, right? And so here you can see the term Pi, and Pi is the pH at which 100% of the positively charged amino acid converts into Zwitter ionic form, right? Zwitter ionic form of amino acid. So isoelectric point can also be defined as the pH at which net charge of the amino acid becomes 0, and at this pH amino acid does not move in response to the application of electric field, right? So in the latter part of my presentation I will explain how you can calculate the isoelectric point of a particular amino acid if you have given the 2 pK a value. Now when you will further increase the concentration of NaOH, then the Zwitter ion will convert into negatively charged amino acid. So this is the negatively charged amino acid by losing one proton by amino group. So at pH 9.60 there is equal concentration of Zwitter ion and negatively charged amino acid. So around this pH, if you will add any strong acid or base, then it will resist any change in its pH value. So around this pH, glycine acts as buffer. So by this way any amino acid acts as buffer. So now I would like to explain about the calculation of isoelectric point from of an amino acid. So Pi can be calculated by taking the average of pK values corresponding to the ionizable groups, right? And so in glycine there are two ionizable groups, that is carboxylic group and amino group, and pK value of carboxyl group is 2.34 and pK value of amino group is 9.60. So Pi would be the sum of 2.34 and 9.60 divided by 2, and if you will solve this equation, then the Pi value would be 5.97. So by this way you can calculate the Pi value of a particular amino acid if you have given their pKa value. Now I would like to talk about the chemical properties of amino acids. So the general reaction of amino acids are mostly due to the presence of two functional groups, namely carboxyl group and amino group. So amine group. So let's first talk about the amino acid when reacts to carboxyl group. So when amino acid reacts with bases, it forms salts, right? And with alcohol it forms different esters. So another reaction of amino acid is that amino acid also undergoes decarboxylation reaction. So after this reaction it forms amines, and amines of many biological importance are used to form by this reaction. Most important example is GABA, that is gamma amino butyric acid, which is a chief inhibitory neurotransmitter in the developmentally mature mammalian central nervous system. So its principal role is in reducing the neuronal excitability throughout the nervous system. Next important reaction of amino acid residues due to carboxylic group is amino acid behaves as base. When it reacts with acid, it forms salts, right? So here when amino acid reacts with ammonia, it forms amide group, right? So for example, when aspartic acid reacts with ammonia, it forms asparagine, and when glutamic acid reacts with ammonia, it forms glutamine. These are the two amino acids which has amide group, right? Now I would like to discuss about the amino acid reaction due to its NH2 group. If in amino acid, amino group behaves as bases, and when it reacts with acid, it forms salts. Next reaction is very important, that is reaction of amino acid with ninhydrin. This reaction helps in the quantitative estimation of amino acids. In this reaction, ninhydrin, which is originally yellow in color, reacts with amino acid and turns deep purple, also known as Rohmann's purple. Here one thing is to be noted, that if proline hydroxyproline reacts with ninhydrin, it forms yellow colored substances. Next reaction is known as transamination reaction, and in this chemical reaction, transfer of amino group from keto acid takes place, which ultimately leads to the formation of new amino acid. So in this reaction, transfer of amino group generally takes place from L-alanine to alpha ketoglutarate, and by the result of that, L-glutamate forms. right? The next reaction is oxidative deamination reaction, and it is form of, and as the name suggests, it is a form of deamination reaction that generates alpha keto acids and other oxidized products from amine containing compounds such as amino acid. It occurs only in liver. So this ammonia generated by the catabolism of amino acid is used to neutralized by urea cycle. So this is all about the physical and chemical properties of amino acids. Regarding this, if you have any question, please feel free. Thanks to all of you, and bye bye.